be happening with respect to the digestive system. It happened to me last night around 9:30 when
It's on the slide. You said it happened to you, so...
No, this event. See, we're talking about the yummy, delicious Panera bagels and the delicious cream cheese. And what might some of you be doing right now? I'm doing it. Some of you are salivating. Or you're thinking, damn, why didn't I eat breakfast? Because my stomach is... That's all part of the digestive process. So your brain and all of that information that you're receiving is actually part of the digestive process, believe it or not, because it's going to prep the rest of the tube for what's to come. So let's continue talking about the basic steps of digestion. So first we have to put it in our mouths. That's ingestion, and that's what we started talking about in class today. I have to want to put it in my mouth, right? Like that bagel sounds absolutely delicious. What else might entice you to put it in your mouth? If it smells good, you have to be drawn to it. So when we talk about the digestive process, there's a portion of this process called the cephalic phase. And that's the part your brain plays in this whole process. So you have to put it in your mouth, and you have to begin with mechanical breakdown. Chew it up, right? Shoot it down, beginning its travels through the tube. The travel from the mouth to the stomach is the esophagus, where we begin, not only mechanical digestion continues, but we begin the process of chemical digestion. What's in the stomach? A bunch of different chemicals, including hydrochloric acid, that are going to help chemically break down your food. And then I have to make it small enough so that I can do what to it? Absorb it. So absorption is part of the digestive process as well. And blood vessels and lymphatic vessels are going to play a big role in the absorption process. Whatever is left that we couldn't break down and we couldn't absorb, we have to do what with? Get rid of. So defecation is a very important part of the digestive process as well. Get rid of the trash, make room for more, right? So all of these things are what we describe as the digestive process in your textbook. And we'll talk about them throughout this chapter. Why is this not? There we go. All right, so in the beginning, we have to propel food down from the mouth into the stomach. A couple of terms we have to know about the food as we prep it for its breakdown. We have to break it down mechanically, chew it. What's the fancy name for chewing? Mastication. And then we have to spit all over it to create a ball of chewed up food and spit. Yum. I know. See, there goes the whole I'm hungry thing. And what do we do? We can't call it a ball of chewed up food and spit. It's called a bolus. And then that bolus has to be sent where? It's going to enter the digestive process. It's going to start the tube travel. So the first part is the esophagus. So when I swallow, food doesn't just go from my mouth into my stomach, does it? And by the way, where is your stomach? Everybody goes, no. It's up right under your, what? Your diaphragm. And sometimes when you eat way too much, you are more aware of where it lives, right? Because you feel like you can't breathe anymore. So peristalsis is the contractual movement that pushes food throughout the entire tube. Not just the esophagus, but when we look at the lining of the tube itself, the tube is the same throughout, except the inner lining might change a bit. and the amount of muscle and the direction that muscle lives in might change a bit as we go down the tube. So when we look at this part of peristalsis, we see it segments out and pushes food down. Ever swallow something or try to eat too fast and feel like, I'm going to die because it gets stuck? And then you can actually feel it moving down your esophagus. That never happened to you? Oh, lucky. Happens to me all the time. So, I think that was an insult. So, peristalsis and segmentation happens, and we're going to find peristalsis happening throughout the digestive system, including the stomach, small intestine, and large intestine. That's how we get everything out, right? What happens if we're getting things out a little bit too quickly? You know where I'm going with that? What's that situation called? Diarrhea. What do you go and take? You take products like Pepto-Bismol, or you take products like Imodium. What are they doing? Slowing peristalsis down. Okay? So, very important part of the whole digestive process. Know all those different steps. So, different chemicals, with the help of receptors to stimulate their release, are going to help with the digestive process. So the brain and central nervous system, as we proved this morning, are part of that process. So external stimuli from the environment can begin prepping the tube for what's to come. So when you drive by Burger King or a burger joint, wow, that smells good. That's prep. That's prepping your tube for what's to come. The stomach, the small intestine, the sight. And this is what happened to me at 9:30 last night when I was watching television and the commercial came on. Looks so good. And all of a sudden, the stomach went. And I said, well, I better go to bed because otherwise I'm going to eat something I shouldn't at 9:30 at night. So the sight of it, the thought of it, oh, I'm going home this weekend and my mother's going to make sauce, homemade meatballs, yum, all part of the process. Because that is going to actually signal later parts of the tube, like your stomach, to start producing what? Gastric juice, which is a combination of that hydrochloric acid and some other chemicals to begin the breakdown. So all of these guys are linked, as we can see under neural reflex pathways, figure 23.4 on page 853 in your textbook. And all of these need that signal to start prepping for what might be coming down the chute with respect to the digestive process. When we look at the digestive organs, there's a lot of that tube. There's an esophagus, there's a stomach, still part of the tube, just a little fatter than the rest of the tube. And then we have 21 feet of small intestine and about 12-ish feet of large intestine. So that's a lot of stuff packed into where? Where is all your alimentary canal hanging out? Most of it's down in the abdominal pelvic region. So this diagram shows us all of the very special coverings associated with the abdominal pelvic region. The mesentery are serous membranes associated with your digestive system. We have another series of membranes called the mesocolon associated with your digestive system. And they're going to help hold all those tubes in place. One of the things I want you to pay very close attention to when you dissect your fetal pigs in the near future is how very complex the mesentery is and how highly vascular the mesentery is. When I look at the small intestine in the fetal pig, if I pick them up and spread them out a bit, I'm going to see the mesentery holding all of those tubes and covering all those tubes together. But what else I'm going to see is tons and tons of little blood vessels. Now, why do I have so many blood vessels in the mesentery holding together my small intestine? The absorption. If I look really, really close, I'm going to see little dots that almost look like grains of sand. What do you think those dots are? Like little lumps. Almost look like little kidney beans. Lymph nodes. Tons of them in the mesentery too. Most of your immune system, those groups of cells, are associated with your digestive system. We also have groups inside, hanging out on the walls, called Peyer's patches. Not as easy to see when you dissect. So there's a very highly vascular and very complex mesentery that's going to help hold in those organs, keep them packed in nicely. And what do you think serous membranes are going to do besides just hold everything together? Yeah, produce fluid, just like we talked about in the heart, just like we talked about in the lungs. So that fluid, pericardial fluid, is going to help all of those moving tubes by decreasing surface tension, decreasing friction. Now, here's the tube. So the tube itself has all of these parts from beginning to end. What's going to change about the tube is this middle portion. So the middle portion we're going to describe as we describe each portion of the tube as being different. But you have to know this general, and where might you see this diagram again? Well, a lab exam, maybe? Mastering, maybe? I don't think it's on this lab exam, is it? Is it? No, I don't think so. We'll have to change that. So, the layers of the tube are the same throughout the tube. What's going to change is how they look, especially the middle one. So the inner layer, well, that inner portion of any tube is called the space or the lumen. So we have a lumen where the food is going to pass along with all of those chemicals that keep squirting on them, on that food as I go. And the lining is going to change depending on where it is, but the lining consists of some sort of epithelial cells. And epithelial cells always have what? Layers. Think. Basement membrane and connective tissue. So we're going to see that as well. That inner layer is called what? It's called the mucosa. Why do you think it's called the mucosa? Another thing I'm going to find dotted throughout all of those epithelial cells is lots of mucus-producing cells as we go down the tube. They're going to help protect the cells that line the tube from being digested from your chemicals that you keep squirting into the tube. So we call it the mucosa. Again, it consists of some sort of epithelial cells, basement membrane, connective tissue, lamina propria, and then some sort of muscle layer. That muscle layer, we're going to find a couple of those, more in some places than others. What's it there for? To squeeze, exactly, peristalsis. So the other thing we're going to find is lots of glands in the submucosa. That's the layer what? Submucosa, under the mucosa. And again, depending on where we are in the tube, we might see different types of glands producing different types of chemicals for the digestive process. After we pass the submucosa, we're going to enter another muscle layer, the muscularis externa. And again, depending where we are in the tube, we might have 1, we might have 2, we might have 3 layers of muscle here. In this particular portion of the tube, wherever it may be, we have 2 layers of muscle, longitudinal and circular. In the stomach, we're going to have 3. On the outside, wrapping everything up and keeping it together, we have that serous membrane, serosa. And then dotted throughout, we're going to find lots of blood vessels, lymphatic vessels, and what are those yellow things? Why do I need nerves in the digestive system? Yeah, because there's muscles there, and who's going to cause the muscles to contract and squeeze the tubes? Nervous system. So we'll find, Yeah. So we'll find them there as well. So know the different layers of the tube. Again, we're going to describe the differences as we go through the text. The enteric nervous system of the alimentary canal is going to help control nerve impulses and control. So the brain is controlling the digestive system as well. All right, in the old days I used to make everybody memorize the teeth. I'm not going to do that anymore. Oh, we're beginning the process. So this is the functional anatomy of the digestive system, and we start right in the mouth. Accessory organs to the digestive system, these guys are going to help the tube do its job by doing what? Starting mechanical digestion and chemical digestion. Chemistry takes better in water, or chemistry works best in water, doesn't it? So I start the process by adding water to it, saliva. So we have the oral or buccal cavity in which the beginning process begins, and they describe that as the mouth. Then the lips and the cheeks are going to help hold everything in place. The tongue's going to help do what? Bounce everything around so I can hit as much of the stuff I put in my mouth with the teeth that are going to grind, mechanical digestion. And then the palate and the uvula are going to help me when I have to do what with that ball of spit and food? Swallow it. So they're going to help push it down the tube in the beginning of the digestive process. When we look at saliva and mechanical digestion, we're going to find that it contains a bunch of different electrolytes and fluid. That's going to help moisten the food, add chemicals to the food so that I can swallow it. Do you ever try to swallow something that's really, really dry? Like every once in a while you try to swallow a pill without, what happens? You might get it down, but then it's like gets stuck right here. And you have to drink, 7 gallons of water to unstick it. Oh, don't do that. That's bad.
I've never done it. But
Don't do it. People think it's easy. Have you ever seen ridiculous people try to eat large amounts of cinnamon? Yeah, no. Don't do that. Anybody? Yeah. You tried? Yeah. Did you throw up? No? It wasn't a pleasant experience, though, I'm guessing. So don't do that. It's not good for you. Saltine crackers. Eat a whole bunch and try to whistle. Yeah, no. 7 in less than a minute.
No water.
Yeah. Sounds easy, doesn't it? Yeah. Yeah, because you don't produce enough saliva. You're going to do some experiments. Well, you already did experiments in lab. I think it was last semester. No, I'm confusing all my experiments. Well, you dried your tongue off. That was a semester, beginning of the semester, right? Yeah, and you couldn't taste it. So it serves a very important purpose. It's going to squirt into the oral or buccal cavity from all around. And who's going to make the saliva? Salivary glands. So the tongue is going to help us with bouncing all the food around so we can chew it or masticate it to create a bolus that we then have to swallow. The term for swallowing is deglutition. So the tongue needs the fluid from saliva so you can taste things. We already talked about all the little taste buds in the beginning of the semester in chapter 15. So that's all review for us on the tongue. The salivary glands are lining the oral or buccal cavity. So we find some under here, parotids. We find some under the tongue, sublinguals, and some on kind of in that mandibular region, submandibular. And all of them are what kinds of glands? Exocrine glands, because they produce their product and then whoosh, shoot it down a little tube. See all the little green things that feed into the mouth? So that saliva can cover the food. And we talked a little bit about gleeking. Yes? Again, when you get all excited about that next meal, you're going to start prepping the tube, and you'll start salivating, getting ready for hopefully what's to come. So those are the salivary glands, and this is some histology on that. So we have teeth. Teeth are going to help us with mastication, helping us grind our food. They are bone, and they are covered with the hardest substance in your body. What is that? It's called enamel. So when you're born, your head's not big enough for all your teeth. So you have what we call what? Yeah, milk teeth, deciduous teeth, your baby teeth. How many of those do we have? 20. 10 on the top, 10 on the bottom. And then as your facial bones form and grow in, you're going to start pushing those old ones out and pushing through your permanent teeth. How many of those do we have-ish? 32-ish. And it seems to be changing as we evolve. The wisdom teeth, or the third set of molars, we're finding that more and more people don't ever grow those. Sometimes you grow 2 sets of them.
Yeah, one of my friends had 2 sets.
My daughter had.
And then she had to go get another set.
Yep, my daughter got hers out and she grew 1 back. We called it the nub. She had to go get the nub out. So, 32-ish for permanent teeth. Tooth is a bone living, full of blood vessels and nerves. Anybody have work done lately? Yeah, you know there's nerves in there, right? So, enamel, very important. Dentin, kind of that bone region that surrounds the nerves and blood vessels. And it is held in, by something called the periodontal ligament. What kind of joint? It's a gomphosis. And see, it's kind of weird. We have the gums, or the gingiva, and then we kind of have a space here before we get a seal. Now, how many of you people, be honest, floss your teeth? How many of you don't? I fell into that category until I started losing some of my gums. You want to be very careful and take care of your teeth. You would be surprised how many complications can arise from you not taking care of your teeth. You think, oh, it's just my teeth, big deal. Well, it's not just for looks, because your mouth is kind of like a sewer. Yeah, you can have complications, cardiovascular complications from issues with your mouth. So it's really important that you keep your mouth healthy. that you brush your teeth, that you floss your teeth. And some of you know my bad habit, but I'll share my bad habit with you. I always never seem to find time to floss my teeth. So do you know where I do it? In the car on the way to work. Hey, I got 40 minutes. What am I going to do? But yeah, I do that too. But find the time to floss your teeth, because it's really important that you do that. And they talk a little bit about some of the different structures of the tooth and some of the problems, homeostatic imbalances, and diseases that are related to tooth and gum disease. So it's not just looking pretty teeth, but it can also harm some of your different organ systems when bacteria gets down in there. The next thing they're talking about is the beginning of the tube. So I've swallowed my bolus after mastication, and it's going to run down this tube. The tube is called what? The esophagus. Now, has all the layers that we talked about. It has the mucosa, the submucosa, the muscularis, and this term adventitia refers to the serosa, the covering on the outside. You can see the lumen running down the middle. Why is it? There's nothing in it. So that tube kind of squishes down. Where does it sit? Where is it physically? Behind the trachea. Remember the C-shaped rings on the trachea? And the esophagus. Now, think back to breakfast this morning. Some of you, that delicious bagel, I hope you chewed it properly to enjoy all of the goodness. By the way, if you're going to eat, enjoy it, right? Chew it really well. The better you can masticate your food, the better the process later on down the tube will happen. But of course, some of us always in a hurry, a couple of chews and down the chute, right? Now, that's about how big it is physically. Was that about how big the piece of bagel you swallowed was? No. So what happens to the tube? Because of that, what kind of tissue do I have lining the tube? Well, there's elastic tissue in that connective tissue area, but important that the mucosa, the epithelial cells that line the mucosa, have to be able to take the tearing of your bagel as it goes down into your stomach, right? So what kind of cells are we going to find here? Stratified squamous epithelial cells, just like we see here, right? Because this has to take a lot of friction as well. If I had simple squamous epithelial cells in my esophagus, you'd swallow once and be done, right? Because you'd rip them all off as they went down. So every time you swallow, you're not only swallowing your delicious Panera bagel, you're swallowing a bit of yourself as well. That's just gross. But it's true. And those cells can be digested and reused in the digestive process. So that's this tube. We have that circular layer and longitudinal layer in the muscularis layer for peristalsis, to push that bolus of food down into the next portion of the tube. The next portion of the tube is a very hostile environment. What is it? The stomach. Now it's really hostile down in the stomach, but it's not so hostile up in the tube. How do we keep the different portions of the tube separate from each other? We have little trap doors in the whole tube. They're called sphincter muscles. So peristalsis is going to squish that food down into the stomach. Once the bolus gets down into the stomach, it gets dropped into an acid bath. We no longer call it a bolus because now it's going to mix with all of those gastric juices. Does anybody know what we call it once it hits the stomach and mixes with all those gastric juices? We call it chyme. So deglutition is swallowing, and that acid bath that we're going to talk about in a moment down there, when it mixes with our bolus, creates chyme. It loosens it up even more and allows all those chemicals to break down that food. Now, that's an acid bath down there. What happens if sometimes the acid makes it past the trap door? Yeah, and they talk under homeostatic imbalances about GERD, gastroesophageal reflux disease. And that's basically, peristalsis keeps going on. If you have problems shutting that trap door well, you might get some washing up of that acid onto your esophagus. Is that good? No, it's like pouring acid all over your cells. If I took acid and poured it on my skin, would that be good? No, it would start to digest it and wear it away. So gastroesophageal reflux disease, very common, is something that can really damage your esophagus, so you have to be careful of that. So, the process of deglutition or swallowing is described in figure 23.13 on page 864. Oh, steps. Maybe? Essay? Maybe? So the tongue's going to help roll things around during the buccal phase of digestion. We have the cephalic phase, me thinking about it. The buccal phase would be what? When I actually stick food in my mouth and start chewing it. And that's going to prep the tube later on down the line. So when you chew or masticate your food, you're prepping the tube down here for what's to come. Do you get the point? Every time something enters the stomach, we're then going to send out chemical messages to prep the tube later on down the line, the small intestine. So we call this the buccal phase or oral phase, and this is when you're actually chewing your food, creating your bolus. And then the pharyngeal esophageal phase is when the food is where? Pharyngeal esophageal phase. Yeah. Where's the pharynx? Behind the mouth. Remember, I shared the pharynx with the respiratory system. Now I'm talking about the oropharynx part of the pharynx. So that's when we have food down there. Now when food gets into that portion, we have to trigger a reflex that's going to stop that food from going where? Down into the trachea. And what was that little trap door called that we discussed? The epiglottis. So when food gets there, we're going to trigger the epiglottis to shut so that food will then enter the back tube, the esophagus. So the gastroesophageal sphincter, and all of these words just describe where everything is. The gastroesophageal sphincter. Where do you think it is?
The end of the esophagus.
Right. The end of the esophagus. The esophagus is in the word, but this is the gastric region that produces gastric juices. So the gastroesophageal sphincter is right before the stomach. Now, it kind of sits, your esophagus feeds into your stomach through your diaphragm. Anybody ever hear of something called a hiatal hernia? What is a hernia? You know, when we talk about all of these different wrappings and surroundings, serous membranes, well, think of a gift or a present all wrapped up in wrapping paper. And you've probably all done this at Christmas because I do it every single time. You finally wrap that gift all up and then something protrudes through the paper on the side. It's like, damn it, got to wrap it again. Well, that's kind of what a hernia is. When the tubes that your serous membranes are surrounding undergo some pressure, or the organ beneath it undergoes some pressure and sort of pops through that surrounding serous membrane. And that can cause kind of a kink in the tube that can cause some issues. So hiatal hernia happens at that place where the esophagus goes through the diaphragm. And depending on how much that kink messes things up and can be a big problem with GERD as well. So, gastroesophageal sphincter muscle opens, food goes into the stomach, we're into the next phase. So the stomach, we're going to find a whole bunch of different muscles in a whole bunch of different directions. Because what's going to happen in the stomach? So the tube widens, but now I have more muscle to have more what? What's that big fancy P word? Peristalsis. So the trapdoors on either side are going to shut because we want to keep the food in there for a certain period of time so that we can get more contraction going and mixing with those chemicals or gastric juices that are going to be produced by the stomach. Another thing we're going to start to see in this portion of the tube is we want tons and tons of surface area because the more chemicals we can secrete all over our food and the more contraction we can have here, the better off I'm going to start breaking things down as we go down the line. So we're going to start to see a bunch of straight things kind of move and wrinkle to increase surface area inside the stomach. That's called rugae. So the mucosa is no longer flat and straight, it's kind of all bunched up to increase the surface area. It's the same thing that will happen in the small intestine. We'll see projections to increase surface area called villi in the small intestine. They're called rugae in the stomach, and of course it's the inner lining, so it's the mucosa. The beginning part is the cardia, and I know you all, when you looked at your slides in lab, said, what the heck are we looking at cardiac for? Isn't the stomach? All right? Some of you haven't had lab yet, but now you'll know the answer to that question because the beginning part of the stomach is called cardia. And that's where that particular tissue came from, cardiac stomach. The next sphincter muscle in line is going to dump into where? Small intestine. And the small intestine, just like the stomach, is names for different portions of it. The beginning portion of the small intestine is called the duodenum. And the sphincter muscle between the stomach and the duodenum is called what? The pyloric sphincter. It's called the pyloric sphincter because it's right beside the pyloric region of what? The stomach. So things are going to hang out in the stomach for a while so I can continue chemical and mechanical digestion. When I look at the lining, the microscopic anatomy of that mucosa, I'm going to see that the cells also create more surface area by bunching up, and that's what we see here at a microscopic level. And they create these little pits called what? Gastric pits. The cells that we find in the gastric pits, we can see them down here, are a group of different cells that have several different jobs. So we have parietal cells down in the pits. We can see them here in this diagram. We have enteroendocrine cells down in the pits. And we also have cells called chief cells. These guys are groups of cells that are going to produce different chemicals to further break things down. These guys here, the purple ones, the chief cells, what do they do for a living? And these are all talked about here on page 866 in your textbook under types of gland cells, because these gastric pits are also sometimes referred to as gastric glands. So they first talk about some cells called mucus neck cells, and those are up further on the line. Why do we want mucus in the gastrointestinal tract or the alimentary canal? So we don't digest ourselves. So it's going to produce at the beginning of the pit mucus to help protect those cells. The next thing they talk about is parietal cells, and those are these little blue guys down here. What do they do for a living? They secrete hydrochloric acid. Why the heck do we do that? We can make hydrochloric acid? Yeah, further break it down. If you take acid, even if you take vinegar, which is a very dilute acid, and put something into it, it's going to break it down. So, the other thing that acid's going to do, produced by the parietal cells, and again, those are little blue guys down there, it's going to help activate some other chemicals that are made by other cells. What are the other cells I'm talking about? The purple cells called the chief cells. And what do the chief cells do for a living? Make pepsin. They make pepsin? No, they make pepsinogen. And when pepsinogen comes in contact with what? Hydrochloric acid, it's going to turn into pepsin. And what's pepsin doing for me? It's going to help me break down proteins. Everybody knows that because they've been studying for their lab exam, right? So, pepsinogen to pepsin has to happen in an acid environment. The parietal cells make hydrochloric acid. The chief cells make pepsinogen. When they come in contact with each other, we get pepsin. Pepsin's going to help me break down proteins. Proteins have to be broken down into what? Simpler form of what? Amino acids. Very good. So proteins are made of amino acids. Remember, when I eat the cheeseburger, that's not my proteins. What do I have to do to the cheeseburger in order to utilize? You have to break it down. So I have to break those amino acids that make the cheeseburger proteins apart so I can then absorb them into the system and use them to make my proteins. Yes? And then, believe it or not, there's some hormone activity going on here. Remember I said this part of the tube might prep for the next thing in line? Yes? If you have gastric bypass surgery, you're going to bypass a lot of the chemical digestion of your food. Absolutely. Yep. So you're going to change this process chemically, radically. And if I can't break things down as much, what else am I not going to be able to do? Absorb as much. So, the next group of cells, the little green guys down at the bottom, also are going to play a role in the process. Those guys are called the enteroendocrine cells. And they're way, way down in the gastric glands because basically their secretions we want to get into the circulatory system to give signals to the rest of the tube. So we see examples of some of the chemicals produced by these guys, basically messengers for the next part of the tube, things like histamines and serotonin and somatostatin. They're going to act as inducers for the next part of the tube. Gastrin is also a very important hormone that is going to be talked about shortly, according to the textbook. Yes? So gastric juice consists of secretions from cells in the gastric pits, all of these guys. That mucosal barrier is extremely important, and sometimes some medications can decrease mucosal production. What happens when I don't have the protection I need? What happens when my cells aren't protected from all of those chemicals that I just talked about? They're going to be broken down, just like the cells that you swallow from your esophagus are broken down. So if I don't have the mucosal protection, I might actually start breaking down my own stomach. Does anybody know what that condition is called? Well, it's called an ulcer. Now, all of that stuff should really stay in the tube, yes? If it starts to leak out into the abdominal pelvic cavity, could that be a problem? It's a big one. So, hormones and paracrines, hormones that act on other cells or signal the next part of the tube that act in digestion, are listed in table 23.1. So some of them, like CCK, cholecystokinin, gastric inhibitory peptide, gastrin, histamine, intestinal gastrin, motilin. I like that one because it stimulates what? Movement. Secretin, serotonin, somatostatin, and vasoactive intestinal peptide are all extremely important chemicals and hormones that aid in the digestive process. Am I going to make you memorize all of these? No. But you should be familiar with them as chemicals as part of the digestive process. So I'm not going to make you memorize who makes them, and I'm not going to make you memorize exactly what they do. Some of them we'll see in some of the diagrams and are a little bit more important than others, especially things like CCK. We'll talk about them a couple of times. But recognize them as chemicals important in this process. Okay? So they talk under homeostatic imbalances on page 869 about ulcers. Way back in my day, we thought that no organism could ever live in the acid bath we've created in our stomach. But guess what? We were wrong. And through research in patients with ulcers, we discovered this lovely little organism. What's its name? H. pylori, Helicobacter pylori. And its association, strong association with ulcer formation. So these guys are acidophiles. They live in very harsh environments like your stomach. And we see them around quite a bit when we see patients with ulcers. So they seem to go hand in hand. Here's one of those awesome diagrams. But this awesome diagram is nice because it ties together all of the different signals associated with the digestive process. So it talks about the cephalic phase, the brain part, and how it ties into prepping the tube down the line. Because everybody kind of signals the next guy in line to get ready for what's to come in the digestive process. So the cephalic phase, we talked about that already, sight and thought of food. Taste receptors are also going to play a role in stimulation of the stomach with the help of that cranial nerve that leaves the fold, cranial nerve 10, the vagus. When the stomach stretches and when those chemicals are released, that's going to signal the next phase, which is called the intestinal phase. That phase is called the gastric phase. And things are going to change a little bit chemically before they trigger the opening of the trap door. That's why I hold stuff in the stomach. Certain foods will stay in the stomach longer than others based on their chemical composition. And what's the other thing that kind of dictates a lot of chemistry going on in the body? pH. So when food that I eat mixes with those gastric juices and the pH changes at certain points, that can also be a signal for the trapdoor to open. For example, when you eat fruits, they don't hang in the stomach as long as when you eat a steak because of the pH and chemical composition of the foods that you eat. So fruits tend to get pushed along faster, open the trapdoor faster, because they're more efficiently broken down than the piece of steak, which tends to hang out in the stomach a little bit longer. All of these guys here also influence what's going on in the digestive system. That's on the list. Anybody get nauseous when you get nervous? There's a connection. Sympathetic nervous system activation overrides parasympathetic control when emotions come into play. Some people actually throw up, reverse peristalsis, when they get really, really nervous. So there is a connection there as well. pH is going to also help the rest of the tube do its job. So this is a nice diagram because it ties everything together for us in all of the different phases of the digestive process. So again, the phases are the cephalic, the gastric, and the intestinal phase. And all of these wonderful chemicals are associated with signaling the tube down the line. So have a general idea of the different phases. Have a general idea of what's going on during the different phases and how they are going to regulate the digestive process on the whole. When we look at page 872, we see this diagram. And this diagram is of the parietal cell. The parietal cell, what did it do for a living? It made hydrochloric acid. Hydrochloric acid is going to help activate what? Pepsinogen to pepsin, breaking down proteins. We do have some absorption into the circulatory system that happens in the stomach. With the help of the parietal cells, we can also absorb some things from the stomach. Some alcohol, for example. Where does it get absorbed into the circulatory system? Does it have to wait till it goes to the small intestine? No, it can go through the stomach. So what if I drink alcohol by itself? You get drunk faster, don't you? How come? There's nothing else for these cells to absorb. Oh, well, I'll get all the alcohol, I'll throw it into the circulatory system. But what if you sit down and have Thanksgiving dinner and the same alcohol? It doesn't affect you as much. How come? Yeah, there's other things these cells are doing in the absorptive process. So it takes longer to absorb that alcohol into your system, and it's going to be at a more even level so your liver can deal with it. So this is peristaltic waves in the stomach, talking about getting food into the stomach. That bolus then is called the chyme once it mixes with gastric juices. The muscular contraction of the stomach is going to grind up that food, peristalsis, and eventually going to open that pyloric sphincter at the end of the stomach and act to push the contents into the small intestine. So they talk about regulation of gastric motility and emptying on page 873 in your textbook. Again, chemical signals, pH, and neural and hormonal signals can control how fast or how slow things move through the tube. So this is a diagram here on page 874, neural and hormonal factors that inhibit gastric emptying. Again, we don't want to push things through too quickly because we want to give them time to break down. So the nervous system has a role in holding on to that food and slowing down the contractile force that will open the pyloric sphincter muscle and dump things into the duodenum. Reverse peristalsis can happen. And what is that fancy name for reverse peristalsis? Emesis or vomiting. And usually that happens when something's not quite right chemically in the body. Now again, nerves can cause reverse peristalsis. But usually it's something that might be toxic or nasty that you put in your mouth that can cause you to reverse peristalsis, get rid of it. So some blood-borne molecules will sense problems and cause reverse peristalsis, and that's described on page 874 under homeostatic imbalances. So the next part of the tube is what? After we pass through the stomach, we go into the small intestine. Now the small intestine is lined with different cells. And the small intestine doesn't like that really, really low pH. So the other thing we have to talk about is some of the accessory organs of the digestive system that are going to help out. Some of the accessory organs to the digestive system we see in this diagram. Liver, play a big role in digestion. What does the liver do for a living? The liver makes bile. Bile is used to do what? Emulsify fat. What's that mean? Not break it down, break it apart. It's like, what the heck is the difference between break it down and break it apart, Maria? What are lipids made of? This is where you're going to go revisit your chemistry brain. What are fats made of? Glycerol and fatty acids. And they're hydrophobic. They hold on tight to each other. So when I eat lipids, they hold on tight to each other. What do I have to do in order to break them down? I have to break them apart. And that's what an emulsifier does. It's going to be easier for me to break it down if I break it apart. You see what I'm saying? So bile is an emulsifier. It's going to help break its hold, the lipid hold on each other, so that enzymes can break it apart. And what are the enzymes that break apart, or break down, I should say, I'm sorry, lipids? Who? Lipase. Yes? So right in the beginning, when we throw things into the small intestine, we see these accessory organs of the digestive system throwing in some secretions to help that process. Bile is not very concentrated when it's made by the liver. The liver makes bile. And there's a little offshoot where bile is going to be concentrated, called the what? The gallbladder. The gallbladder is where I'm going to concentrate bile and then eventually expel it into the small intestine. Anybody ever make rock candy? How do you make rock candy? I take water and sugar, and more sugar, and more sugar. And I have to heat it up to actually dissolve the sugar in that really concentrated solution, right? And then what do? Stick a string down in the jar. And what's going to happen? Yeah, it's going to start to crystallize, go back to its solid form out of its liquid when it's highly concentrated like that. And it's going to crystallize around the string, rock candy.
While it's still liquid?
In the liquid. So some of the molecules are going to go back to solid, and they're going to hang on the string. So when you concentrate solutions, you have the chance of them going back to their solid form. And what might that be in the gallbladder? Gallstones. And that might be a problem, because the stones might get to a place where they're going to disrupt the secretions. And that could be an issue, right? So sometimes we have to get rid of the gallbladder and its stones that have formed. You just tended to form those crystals. So they get rid of the gallbladder. It's okay, because the liver makes bile, so you're going to still have bile to help break the hold of lipids. But now it's not going to be what? It's It's not going to be concentrated. It's kind of like the two different laundry soaps that back in the day when liquid soap first came out, big huge jugs, and how much did you have to put in the wash to clean your clothes? Like a big cup. Nowadays, you're doing the same thing. You're not doing anything different, except you only have to add this much. How come? Because we sucked all the excess water out and concentrated it. That's what the gallbladder does for the liver. It shares a little duct with another exocrine gland associated with the digestive system called the what? Pancreas. We already met the pancreas when we talked about the endocrine system. It also has a digestive function as well. So it's going to make a bunch of different enzymes to help food digestion, as well as the hormones that it makes to help regulate some of these different molecules as well. So the inside of the small intestine is discussed under gross anatomy and histology, microscopic anatomy. So the gross anatomy of the small intestine, 21 feet, long tube. If I pulled mine out here, I'd smack into the column. We have a little valve or sphincter muscle called the ileocecal valve. It's going to help things stay put. The duodenum is the beginning, the jejunum, and then the ileum as we move through the tube. Microscopic, different than the gastric, right? So mucosa just changed again. You're still going to have all the same layers of the tube, but now it looks different on the inside. What happened to the gastric pits? They're not pits anymore. They came out. What are they? They're villi. They're not microvilli because I don't need my microscope to see them. So they're villi. So they have finger-like projections, and we see it here, associated with the lining or mucosa of the small intestine. Why do you think I have these? Increased surface area. More cells are going to come in contact with more stuff in the tube. The other thing I'm going to find running up between all these little finger-like villi projections is lots and lots of what? Blood vessels and lymphatic vessels. Why lymphatic vessels? Yep, we talked about it last class. Absorption of fat, chylomicrons. Those are going to get absorbed into the lacteals, the portion of the lymphatic system associated with the villi. And where are they going to dump those lipids off? Yeah, up in the thoracic duct in the neck region. Yes? Okay. The cells that line the villi also have villi. What are they called? Microvilli. So if I look at the cells themselves, and these are simple what? No squamous. Columnar, simple columnar. Whole bunch of goblet cells in there. Why do I have a whole bunch of goblet cells in there too? Mucus. Got to produce the mucus to move everything down the tube. Protect that lining. I'm going to see that has villi as well. We call them microvilli. And if I look at this real close and use my imagination, it almost looks like the head of a brush. So the lining of the small intestine is also sometimes referred to as the brush border. These cells also make enzymes to help in the digestive process. Guess what we call that group of enzymes? The brush border enzymes. Who makes them? The small intestine. So we look at a picture of the brush border and the microvilli on the cell. So this is the top of an actual cell. So this is the what? This is the liver. Huge. How big is your liver? That big. Yeah. Yahweh. So you see your diaphragm? Guess what lines the whole inside of your diaphragm? Your liver. So you go like that, that's your liver. And there's your liver here. Your liver, very highly vascular, big important organ. For the digestive system, it makes bile. But what else? We've talked about it a couple of different times. It gets rid of old blood cells. It detoxifies blood, filters blood. It's also a stop for all those lipids before they hang out into the circulatory system for a little bit more processing as well. So there's a lot going on in the liver, and there's a lot of it. Yeah, well, not the only, but yes. So if I get a liver transplant, do I have to get the whole thing? I can donate my part of my liver and still survive, right? To somebody else. Did you know that? What's it going to do? It's going to regenerate itself. What's it going to do? The piece that I gave to my buddy, what's it going to do? It's going to make more. Okay? So yes, liver is very complex. When we look at the microscopic anatomy of the liver, everything's kind of little compartments that we see. If we look at the top, we see the vascular portion in the central vein region, and then it sort of looks like honeycombs. One of the things I forgot to do in lab was put out the slide of the liver, so you won't have that on your lab exam. But you should be familiar with the microscopic anatomy of the liver. And it creates these little compartments surrounded by connective tissue and lots of blood vessels. So within this little compartment, we're going to find tons and tons of different cells. Some are going to be breaking down old red blood cells. There's a whole bunch of macrophages that hang out in here called Kupffer cells with a K. And those are the cells that are going to help get rid of debris. That's what we call the macrophages in liver, Kupffer cells. And then we also find cells that produce green stuff. What's that? Bile. And they're going to be transported to the who? Gallbladder for concentration. Or if I had to get rid of my gallbladder, my bile is going to get thrown right into the small intestine through this portal system. So we call it a portal triad because we are transporting bile. We also have venous and arterial blood running through the liver as well. So tons and tons of blood vessels. Very, very vascular, very big job. And with respect to the digestive system, the liver is important for the production of bile. It also is going to help filter out any nasty things that we eat. So it's going to help detoxify things in the beginning of the absorption process. The hepatic portal system, with respect to the circulatory system, is going to feed in from all of the mesentery of that small intestine into the liver first before blood makes it through the rest of the body. If you don't believe me, go back to chapter 19 and look at the stuff I didn't make you memorize. But you can see where the hepatic portal system makes its connections to the rest of the body. So when we absorb food, it's got to go through the liver first before it makes it to the rest of the circulatory system. And that's what the hepatic portal system is. So some of the homeostatic imbalances of the liver can create problems with chemical balance in the body. Hepatitis is a nasty little virus that wreaks havoc with liver and can cause it to be dysfunctional. One of the things the liver does is process those broken down red blood cells. And one of the things that the waste products that the liver helps us handle is something called bilirubin. When somebody is having problems with liver function, that bilirubin can actually build up in their blood plasma. You know what color bilirubin is? It's yellow. And that can result in something called jaundice. Cirrhosis, portal hypertension, all described in your textbook, all dysfunction of the liver. So the liver is really, really, really important. And you want to take care of your liver. How do you take care of your liver? Don't go to Jamaica all the time. Don't apply too many toxins to the process. Toxins in the form of alcohol could be one, but toxins in the form of Twinkies can be one as well. Because when you eat foods that are full of toxins, your liver has to process them. Pesticides, process. Chemicals that are added to your food that don't normally belong in your body, process. Who likes blue Gatorade? Come on, raise your hand. Anything blue. Do you know what makes it blue? Chemicals. Chemicals made from ground up bugs. Chemicals made from, go look it up, go Google this stuff, I'm not kidding. How about those red M&Ms? Yum! What makes them those beautiful colors? Petroleum-based products. That's gasoline, my friends. And they make these beautiful colors in your food.
Raspberry flavoring,
Yeah, the blue stuff.
and strawberry flavoring, and some vanilla ice cream to sweeten it is beaver anal gland.
Go look this stuff up. We're not kidding. So, what might help your liver will stick with food in its what? In its natural form. Because every time you eat something that has all of those additives and chemicals and preservatives, guess who's going to deal with that stuff? Your liver.
I saw this picture on Facebook, and it was like this fitness person, and they posted this picture, and it showed food from like Wendy's, Burger King, and McDonald's, and it was like 2 years old, and it looked like it was brand new food. Like, didn't even like mold or anything, and like bugs wouldn't even touch it. Like, it was
When
just
bugs won't touch it,
that's bad.
don't eat it. If you can't pronounce it, don't eat it.
Takes what, 6 months to break down the fats, something like that.
I don't know about that, but yeah, it's those foods are taxing your liver. Not just alcohol, but those things too. Toxins in your environment tax your liver. Toxins in your water tax your liver. These are a close look at some of the different cells that we see in another little friend of ours. And this is our pancreas. So we revisit the pancreas when we talk about the digestive system and its role, the role that it plays in the digestive process with chemical composition. Also, don't forget to take a quick look at what bile is. It's a salt-like material, and it's an emulsifier. So the gallbladder is really important, and the pancreas is talked about next. The group of chemicals produced by the pancreas is called pancreatic juices and contain chemicals that are going to break down further some of the molecules that we need to break down. So we have lipase that is produced by the pancreas. We have trypsin that is produced by the pancreas. We have carboxypeptidases that are produced by the pancreas. And enteropeptidase within the small intestine is going to trigger all this release. You can see this on page 883 in your textbook under the composition of pancreatic juices. Again, this is going to help regulate the small intestine movement of food through it. And this is an example of some of the different chemical activity that's going to happen here in the small intestine. One of the things you have to remember is the small intestine doesn't like that really acidic pH. So the secretions that we see from the pancreas also are going to help to neutralize that very acidic chyme that comes from the stomach. One of the places where ulcers are very common, if you're going to get one, does anybody know? One of the places where ulcers in the digestive system are very common is the very beginning up here. They're called duodenal ulcers. Okay, because sometimes we have decreased mucus protection right at that beginning before we get the secretions that neutralize that acidic chyme. Make sense? So, woo! This looks like an essay question ready to happen. Mechanisms promoting secretion and release of bile and pancreatic juices. So again, the entry of chyme from the stomach after it passes that sphincter muscle is going to cue the release of those accessory organ secretions. And that is going to help signal the rest of the tube. Cholecystokinin is the one you should remember at the beginning of the small intestine. And that in the bloodstream is going to help signal the release of some of these secretions along with some of the important events that are going to happen later on down the line. So CCK and secretin are hormones that we see here that get into the bloodstream that are going to help prep the rest of the tube at this portion of the digestive process. This diagram on page 885 is an overview of all the different functions of the gastrointestinal tract. So we see the mouth and the esophagus and the associated organs there, the pharynx, which leads to the esophagus, the stomach, what happens there. This is one of those really good tables that's going to make everything small for you. So use it to your benefit. Small intestine, and then the large intestine. So the digestive process in the small intestine, we're going to continue with motility. We're going to continue breakdown with the brush border enzymes, and we will pick up with the digestive process in the small intestine on Thursday. Let's do a quick clicker question so I can take attendance. And we already did that one. So the majority of absorption occurs where? I can't see it. Everybody click. 5, 4, 3, 2, excuse me, 1. What's the answer? The small intestine. Want to do one more? The majority of blank occurs in the mouth, stomach, and small intestine. Mouth. 5, 4, 3, 2, 1. What's the answer? Mechanical breakdown. Adios. See you on Thursday.